All right, so we're doing Big Lab 8 for biology here, and this is where we have these simulations where we exchange genes, and so we're going through a few things here today. So in part 1, I'm going to read through case 1. Now I have 2 classes, AP Bio, so first period, actually let me close that door. First period is going to use your DNA for case study 4, which is genetic drift, since we have a very small class, and we would expect more changes in the DNA, even with random mating because of that small population. And then 5th period is going to use the data for case study 1 from first period. And so because they have a much bigger class, and hopefully it's not affected as much since they have a bigger class and a larger exchange of DNA. So let me read through case study 1 here just so you know what's going on, because case study 1 and case study 4 are basically the same, except in case study 4 you're supposed to break up into a smaller population, and since we already have a smaller population in here, we only have 10 people, then that's why we're doing case study 4 in 5th period. All right, so basically what happens here in these scenarios, the entire class represents a breeding population, okay? The class will simulate a population of randomly mating heterozygous individuals with an initial gene frequency of 0.5 for the dominant allele, big A, and 0.5 for the recessive allele, little a, which would mean the genotypic frequencies would be 0.25, 0.5, and 0.25, okay? So basically what we're starting out with here is we've got 2 little a's, and we've got 2 big A's. Okay, so all these together, we're heterozygous individuals for this gene trait. Okay, and so we're going to use these cards to help exchange DNA. Okay, and so this is essentially simulating meiosis, where you start out with 1 cell, and then you wind up with like 4 sperm, and so you would wind up with your 4 different variances. And you're going to randomly kind of shuffle these up, and then you'll see which one of those traits gets passed on to your offspring. Okay? So that's what the 4 pieces of paper are. Now, your initial genotype is big A. Little a, record this in the data page. Each member of the class will receive 4 cards. So you've got your 4 cards. 2 cards have big A's on them. 2 cards have little a's on them. They represent the products of meiosis. So you guys, in my particular class here, I know I'm reading through case study 1, but you guys are going to write this data down where it says case study 4, because this is a small population and it's basically the same rules for case study 1 as case study 4, except case study 4 is supposed to be a smaller population. Does that make sense? So in case study 4, go ahead and write down your initial class frequencies, which were 0.25, 0.5, and 0.25. And then your P and Q frequency, since we have an equal number of big A's and little a's, is going to be 0.5 and 0.5. Good. Okay, so your initial genotype is the initial genotype we're starting out with.
25.
Big A and little a. Good. So genotype are the genes that you have. So we're all starting out with big A, little a. And then we're going to exchange DNA a few times. You can see the data sheet here. And that's where the different phenotypes are going to come in. At the end, I'll give y'all someone's data so that y'all can share this. So for those of y'all in my class that are actually doing this at home. So basically what you're going to do is you're going to turn the 4 cards over so you don't see them. You're going to shuffle them up, and you're going to go find a partner. So I've got it split up where we got 5 people on this side and 5 people on this side of the room. And so I'll say go find your, your mating partner. And one person will come from over here to this side of the room. You will shuffle your cards. You will put down one that you pick out randomly, and then they will pick out one and put down randomly. Now, so one of you needs to decide, like, I'm gonna assume the first offspring. So let's say, Walker comes over here and he mates with Nathan, and the first baby that they come up with is little a. So Walker's like, I'm going to be that one. So basically you're going to write down for your F1 generation, your offspring is little a. Okay? So you're going to then become little a. So you're only going to have 2 little a's the next time that you go and mate with someone. Does that make sense? So you're becoming the offspring each time. So, and then you'll put all your stuff back in there, you'll reshuffle, and then Nathan has to have an offspring too. So you've got to, randomly put down 2 more. He puts down 1, you put down 1, and then he will become that second offspring that y'all produce. And then he'll write down that offspring, and then that's what, like let's say you put down 2 big A's. So then he will assume the genotype of big A when he goes to mate next. Does that make sense? So you're becoming the offspring, and you're becoming the genes of those offspring the next time you randomly mate with someone. Okay? So you still need your 4 cards as we work through this. So probably the best way to do this is to just do this. So everybody's got their 4 cards. If you're on this side of the room, you're going to go find a random mate on this side of the room. You're going to produce 2 offspring. One of you will become one of those offspring. One of you will become the other offspring. You'll write that down under the F1 generation on that data table under simulation 4, and then you'll go sit back down. So we're going to do this in a very orderly manner. I found out that if I just let people loose mating randomly, that somebody's going to be on their 4th generation over here, and then somebody over here will be on their 2nd generation, and then it will never work out. So we're all going to do this at the same time. So if you guys will, let's do it this way. If you're on this side of the room, come over here, find somebody to mate with, and lay down your cards, write down your F1 generation, and then go sit back down. Okay? No touching or heavy petting, please, though. This is just a simulation. Okay, so once we get through this entire simulation, you're going to figure out, so we've got a certain number of different genotypes from each one, the big A, little a, and little a. And then you've got to figure out the P and Q frequency, the allele frequency in the generation. Remember, we started out with half and half. Everybody was heterozygous, so we had a 50% dominant and recessive allele frequency. Okay? So for you guys in this class, since this is 5th period, you're going to use the data from my 1st period class, okay, in that first simulation. So let's go ahead and write that down. So at the end of the first simulation, we wound up with 5 people who were big A. We wound up with 9 people who were big A, little a. And we wound up with 2 that were little a. Now remember, we started out with everybody was big A, okay? And we had a 50% of recessive and dominant alleles, okay? So now we need to use this data to figure out our allele frequency, the dominant and recessive allele. And remember, P is going to be your dominant, so that'll be like big A, and Q is going to be your recessive, which will be like the little a frequency, okay? So we're not going to use Hardy-Weinberg to figure out that allele frequency. We're just going to count alleles and then do the percentage. So if you've got big A, how many, and we've got 5 people who are big A, how many big A's is that? 10, because have 2 of these. So that's 10 big A's. If we have 9 people who are big A, little a, how many big A's is that? 9. So we have 19 big A's out of 32 possible because we had 16 people in the class. Does that make sense? 16 people, 32 alleles. So 19 of those 32 alleles were big A alleles, okay? So you can use that information to figure out the percentage of P. So what the P, the dominant allele frequency is. And then the same thing, we've got 9 little a's. We had 2 people that were little a, which would make 4 little a's there. And so 9 little a's plus 4 little a's. 13 out of the 32 alleles were for a little a. And so you will use that percentage to determine your Q frequency, okay? So both of these simulations, the ones you guys just did and the ones they did, were all just random mating. There was no natural selection, no trait that was selected for. And so you would expect with random mating what would you expect to happen to the allele frequency? Would you expect it to change or stay about the same? Stay about the same. Now, if you remember though, there's about 5 different requirements in order to maintain genetic equilibrium, or basically no evolution. And one of those that we don't have, especially in here, and we really didn't have with 16 people in first period, is what?
Large population.
Large population. And so the smaller the population, the more likely you are to see a variation or change in allele frequency. And we call that, simulation 4, genetic drift. Good. So let's go through and look at what we got in simulation 4 and see if it worked out. Sometimes it works out and sometimes it doesn't. At the end of simulation 4, how many of you guys wound up with big A, big A? Raise your hand, sorry. 1, 2, 3, okay, not you, right? So we had 3 people in this class who wound up with big A, big A, okay? We got 10 people in here. How many of y'all wound up with big A, little a? 1, 2, 3, 4, 5, 6. And how many had little a, little a? Just had 1, okay. So there's nothing wrong with you, Katie, don't worry. So this is our, and you're gonna use that data to figure out your P and Q frequency, okay? So we'll see if it's different in a smaller class. Maybe it looks about similar, to be honest with you. And so maybe it's a little bit different, I don't know. But typically, the more generations you go through, the more differences you would expect to see in a smaller population, okay? So that's simulation 1 and simulation 4, an explanation of how you do those. Now, we're gonna work through simulation 2 really quickly. Simulation 2 is a case study. If you guys will turn back, we'll go through and do all these simulations, and then you can go back and do the calculations. So in simulation 2, we're applying a little natural selection here. So in a natural environment, not all genotypes have the same survival rate. So some are better than others in that environment. An example is sickle cell anemia. This disease is caused by mutation. For individuals who are homozygous for sickle cell, many do not survive to reach reproductive maturity. For this simulation, you're gonna, now, they call this homozygous recessive. I call sickle cell a codominant trait. So just to clarify for the biology part of that, if you inherit sickle cell alleles from both parents and you have sickle cell anemia or sickle cell disease, whatever you want to call it, then your cells, most all of them are going to be sickled. They're going to be funny shaped because the proteins are messed up. If you have normal blood cell genes, you're going to have normal round concave blood cells, okay? If you inherit a sickle cell gene from one parent and you inherit a normal blood cell gene from another parent, you're going to have some sickle cell and some normal blood cells. So it's really more of a codominant trait. It's just, I think for the purposes we're doing here, they're calling it recessive to kind of fit into our P's and Q's. So just to clarify, codominant trait, but we're kind of treating it like a dominant recessive here. So basically what we're going to do is we're going to say the sickle cell is a recessive trait. It's going to be our little letter. And for this simulation, you will assume that homozygous recessive individuals never survive. So basically, if you're at little a, that baby's going to die. And so you still need a baby, so what are you going to have to do? What do they do back in the day? What do they do in places right now if you have lots of babies that die? Keep having babies, there you go. So we're going to, if your baby dies, you're going to make another baby until you get one that survives. And so that you can become that offspring in the next, in the next generation. Does that make sense? So if you come up with little a, you're just going to have to go back to the drawing board, make another baby until you get one that survives. And then we're going to go through our 5 generations the same way on this, and we're going to see what things look like at the end. Now, we're all still starting out heterozygous here. You've all got big A's and you've got little a's. So everybody, the allele frequency is 50-50. But this recessive trait, as they're calling it, is getting selected against. So no little a's are survivors. You can still have little a's in the population, but it would have to be heterozygous little a's. Does that make sense? So let's work through our simulation and those 5 generations. Everybody's starting out big A, little a. We're going to do the same thing. So in simulation 2, we wound up with 5 individuals that were big A, and 5 individuals that were big A, little a. So about half and half. after we went through 5 generations. So the trait was selected against was little a, obviously none of those survived, and then the other traits were equally as viable, and so we wound up with an even split, which is a good, that was fantastic. Now, in the third simulation that we go through, where did my paper go? Who cares? So, in the third simulation, there it is. We're going to go through, this one is a little bit different. So basically, one thing you need to know about sickle cell is it's called heterozygous advantage is the term that's used. And so we're going to switch one thing up. So little a's, they're still going to die, okay? Out there, not everybody with sickle cell anemia dies, so don't worry. This is just for bio. So everybody with little a's, it's still going to die, and you're going to have to reproduce, okay? But let's pretend we're all hanging out on the jungles of Africa here. And if you look at people that are sickle cell carriers and people that don't have it at all, which ones are more likely to survive in the Congo jungle? Carriers, because they have some malarial resistance to them. And malaria is prevalent, and it can often be deadly. I think roughly about 500,000 people die in sub-Saharan Africa each year from that. So it's beneficial. So how do we show that it's beneficial in these phenotypes, in these genotypes as we're passing on these traits? That's a great question, Mr. Reynolds. So this is what we're going to do. If you land on big A, okay, then you're going to flip a coin. And when you flip that coin, where is it? I must have erased it. If you land on heads, you're going to die. And if you die, that means you died from malaria before you were old enough to make a baby. So if it lands on heads, you're going to die from malaria. You're going to have to make another baby. If you land on tails, then you survive, and then you can live to pass on your traits another day. Okay? Does that make sense? So little a, it's going to die. You got to make another baby. If it lands on big A, you're going to have to flip a coin. I've got coins over here on my desk, so you can have one. You're going to flip a coin, and if you land on big A, and it lands on heads, then you're going to die, and you got to make another baby. Okay? Let's say you land on big A again. Well, let's say you make another baby, and it's big A. Well, you're going to have to flip a coin again, and if it lands on tails, then you survive. If it lands on heads, you're going to have to make another baby. Okay? So we had a lot of babies that had to be made this morning until we got to where we needed to go. Sometimes, but that's okay. Okay, so that's simulation number 3. All right, we've just completed simulation 3. We went through 6 rounds. We have time for 10. That's okay. And let's find out the number of different genotypes that were left after 6 rounds of that. So raise your hand if you wound up with big A at the end. 1, 2, 3. Okay. And then big A, little a, we have 7. 1, 2, 3, 4, 5, 6, 7. Okay. And then little a, all those died. So that's actually way better than what we had first period, because first period had way more big A's, which I was surprised, but sometimes that happens. And so this is kind of what you would expect to happen when you have, half the individuals dying because of that selection, natural selection for malaria. So you guys are going to go through, you're going to figure out your allele frequencies, the big A, little a frequencies from these, and then you've got a few questions to answer. So the questions are a little bit iffy. Let's turn and look on page 93 here real quick. We're going to modify this. They're asking a few questions that I'm not really sure what they even mean right here. So we're going to modify this just a tad to make sense for what we were doing. So in number 1 on 93, and so this is actually referring to simulation 1, okay, or case study 1. Like I said, I gave you data from this from first period. What does the Hardy-Weinberg equation predict for the new P and Q? We remember P and Q started out at 50-50, okay? Really, all I want you to elaborate on and discuss is, did the allele frequency change with random mating? Okay, so all we had in simulation 1 was random mating. Nothing was selected for. Did the allele frequency change with the random mating? Okay, and then number 2, all I want you to discuss is, why did it change? Okay, and number 3, you can mark it out because I don't know what they're talking about. All right. So, did it change? Number one, why did it change? And there, you should have a very good reason for that. I'm pretty sure we already discussed it. And then you're going to answer the questions on pretty much the rest of the questions on the other two simulations, and actually the third one as well. So you can get with your partners, y'all can work through that. This lab, the Teddy part and this part is going to be due on Friday. Okay. Tomorrow we will be working a lot on microevolution part 3 and macro part 1.